I'm Fernanda. I am currently doing a postdoc with Martin at DTU, but during my PhD, which was in Brazil, I did some collaboration. And then, some months ago, one of the chapters of my PhD was published in this paper. So I'm going to talk about this work, which has a title more like a conclusion. No sign of homogenization in reef fish communities across an ecological transition zone exposed to warming. So I'm going to change a little bit the topic that we had the presentations before. So it's not going to be about complex models or like high seas fish, but it will be about these colorful creatures that I like a lot. So just to start, so why homogenization matters in ecosystem assessments? All of you are working with this approach, so you need a lot of indicators of the status of the ecosystems, and homogenization can provide one of these status. But what is homogenization? So by definition, homogenization, it's when two communities, two biotas become more similar. Normally over time because of warming. So it's basically the movement of a generalist species, like increasing in abundance, expanding their distribution, and making the communities more like similar over time. So homogenization can be assessed by the different metrics. One of them is the similarity. In this year is just a temporal trend of, I guess I can use these. Yep. So just a temporal series showing how the Jacquard's similarity is increasing over time. So this means in this case is similarity, so higher values represent more similar communities over time. And this has been reported for different systems, terrestrial, marine, and so on. So this we are thinking about the species that are present, just the identity of species. But we can also think about the traits of the species that are in the communities. So what happens is more or less like a funnel. So you have some communities more diverse in the beginning, let's say in the time series, with different traits, performing different functions. But then over time, because of the increase of generalists and loss of specialist species, the number of traits they decrease, and then the community change and become more generalist dominated. But back to the question why homogenization matter. So it actually provides a nice indicator of a loss of rare or specialist species in such an area. Also the expansion of tolerant or generalist species, and also normally it provides an index of declining functional roles. So as I said, many studies address this homogenization in relation to global warming, basically because the species, when they are under warming conditions, they tend to migrate towards different habitats. They colonize new habitats and then expand their range distributions because they have traits that allow them to survive in these new environments. So and this is not new for you. So the global temperature is increasing. We also have other human pressures happening, such as fishing, pollution, and so on. But then assessing if the communities are getting similar or dissimilar can provide early warning indicator, and you are able to identify hotspots of change, and then adapt management plan. So we used the Southwestern Atlantic as a case study to test the hypothesis of homogenization. First of all, because this area, so we are in the South Brazil, and then there is a big island here, a very nice one, Florianopolis, and then we have small islands around. But when we look at the temperature, the sea surface temperature of the region, we can see a gradient from the warm waters in the north and cold waters in the south. So this it's basically a transition zone. So we have a gradient of different conditions. And we have fish communities in each of these islands. And then there is a monitoring program in the university that I was doing my PhD, where we, in every summer, we go to the sea, we dive, and then we count the fish that are there. We estimate the abundance and length and so on. And this happened since 2007. So we took this temporal series to test if the communities here, actually here in the north part, if they are getting similar to the south part. That was the hypothesis. And also because we normally have like this gradient in temperature, like because it is the condition there, but also through time, if you look at the trends of temperatures, they are also increasing. So there is kind of a warming pressure happening there. So we hypothesize that they will become similar over time. So yeah, how did we do that? So first we took like the samples. So we had like different under-visual censuses in all of these islands. And as you can imagine, the number of censuses was not equal in all of them, so it was quite unbalanced. And then we did some magic to avoid this bias using species accumulation curve, but I'm not going into much detail on this. But then we try to handle the bias. Then after that, we had a list of species present in each island, and then we did trait compilation for all of the species. So we collected data regarding the diet, regarding the type of the caudal fin, the mouth position, the body shape, and so on, for all of the species. And with these values, we were able to build the similarity matrix and then calculate the index that we wanted. So it was alpha and beta diversity. We used two techniques because of the nature of the data, so convex Hu and hypervolumes. And then we estimate how the taxonomic and trait diversity changes over time using the occurrence data, so just presence, absence, and also abundance. So there was kind of two steps. First, we computed the metrics for each region isolated, so for north and for south. And then to really test the homogenization, we compared both regions at the same time, north and south together in the first year, second, and so on. And then it was a pairwise analysis. And then with this, we were able to see the patterns and check the homogenization. The results that we had, so first, we could actually see some temporal variations in species and traits for both groups of the islands, for north and for south. So here what you are seeing, it's looking at 4 dimensions, 4 different aspects of beta diversity. So in this case, it's a dissimilarity. So values close to 0, communities are totally similar. Close to 1, they are totally dissimilar. And so we also calculated for taxonomic and trait, so the 2 colorful lines. And then in the first panel is using presence, absence, and the second one, abundance. I just wanted to highlight that this is temporal distance, so it's not each year individually. It's always referred to the first one, so the comparison of the third year to the first. So basically what we are seeing for the north, there's no strong pattern, so there is kind of increase in taxonomic dissimilarity for north when you account for presence absence, but the traits, they are relatively stable, so the functions that the species are performing there, they are quite the same. But in contrast, the south part, we can see like a more pronounced change, especially in accounting for abundance for taxonomic. So in the south, when you just compare isolated, this region is probably losing species and becoming more dissimilar over time. While the traits, they have some kind of trend, but not very, very strong. So okay, after that we have the trends for each, but then to look into the biological data, we wanted to check what are the species that are driving these patterns. So we performed a simple analysis. It's a simple analysis that calculates the contribution of each species to the total variation. So in this y-axis is the cumulative contribution of the most cumulative contributions of all species. So it's ranked like from the highest contributors to the lowest one for north and for south. And then the color that you are seeing in these box plots is a value of distinctiveness that we calculate. So what is this value? It's actually a value of how different the species are in terms of traits compared to the overall communities. So if a species has a higher distinctiveness value, it means that this species has like a very different traits, such as like a totally different, like a mouth, or perform a different hole in the ecosystem and so on. And when this value is low, it means that it's mostly the dominant species and kind of normal species that are common traits. And then what we see in our results is that the most contributors to the variation in these islands are actually the common species, the most abundant species. So it's not like a super different or very exotic or rare species. No, they are like the most common. Oh no, no. Yep. Okay. There we go. Okay, then when we compared, because now I presented it individually, the islands for North and South, but then when we compare together, then we have again a present substance, just the occurrence of species and weighted by abundance. And then surprisingly, we didn't see any sign of homogenization. So the communities are not getting similar over time. Instead, they are becoming dissimilar, especially when we account for abundance and the richness of these species, because we can see that the biodiversity values are increasing towards 1, while the traits are remaining quite stable. But yeah, it was totally different than we expected. It was probably we have some theories about that. It's probably because of the South Islands, they are losing some species, not some species, but not in terms of extinction, but they are decreasing in abundance and making different than the North. Yeah, so as a take-home message, we highlighted multiple metrics of alpha diversity, beta diversity, weighted or not by abundance, is also considering traits of species. It's important to understand the changes in diversity and the structure of a system, because as I showed, for some metrics there was no change at all, but when you include the other variables like other factors, then you can see some patterns. So yeah, again, there was no homogenization across this transition zone, despite the general warming trend. This doesn't mean that the communities there are totally safe or okay in relation to climate change, but they have some sort of resilience so far. And then when we compare the total richness of the Southwestern Atlantic or Brazilian coast, of course, is very different than Caribbean, than Australia. But then we highlight that the high trait redundancy maintain a range of ecological processes due to few functional entities. So we have less species there, but they are performing well. So they are doing like their jobs. And yeah, and then lastly, just highlighting the significance of preservation of the trait structures of the community, not just looking to species richness, the identity, to predict potential responses to disturbances. So that's it from my side. Thank you. Obrigada.
Okay. Thank you very much, Fernanda. Do we have any questions for Fernanda? Guillaume.
Thank you very much. Nice results. I would like to just to share a comment that even the results are very nice and important. I was not very surprised of this, I would say, temporal decay of similarity. Because we have been also have done a similar study in zooplankton species also in places that were not influenced by anthropogenic, but yes, with the warming. And we have also seen this pattern of, I would say, temporal decay of similarity. And I think that this assumption of homogenization maybe is not very general. So because you have climate change that is imposing this driver of ecological drift in the communities, but also you have the ecological drift itself of the communities that tend to drift the similarity in communities itself that also is running independently that you have an anthropogenic, even if you don't have anthropogenic or climate change driver. So to me, it was not surprising. So.
Are there any other questions or comments? Elliot?
Yeah, I was wondering if how often you manage to get out to the islands. It seems like a really big field expedition for your research group, but you've got this kind of trend happening on annual scales. But I would imagine the South has more variability in intra-annually. So is there any what would be the dynamics with the kind of disruption of like seasonal fluctuations in temperature relative to a more stable in the north? Do you think that has a role in driving the homogeneity?
It probably has an influence for sure, especially in the south part. But then I think the main issue is that we don't collect data during winter, simply because it's too cold. I mean, weird to say here in Denmark, but yeah, kind of complaining about 20 degrees. But yeah, so we don't have data to compare both seasons. I assume that in the south it could be stronger because then it's the south, and then the winters, like it can get a little bit more intense. But not that much, because in total, like the coastal area, if you measure like the first island to the last one is around 47 kilometers, so it's a very small scale. But they are not. It is a nice thing that we like to try. Also taking into account recruitment, but then to get data for that is a bit tricky. And also because we have to go to the field, and then we have to be familiar with the species. We have to count. Then there's no like a gear that we put, and then yeah, it could be definitely something.
Thanks.
More questions? I've got a question myself. I was wondering, this is very relevant for the management of the region, but taking into account that your results are overall positive, did you compare your results to policymakers, or would it be not a very good idea because it means like don't do much because the system is resilient at the moment? Mm-hmm. What's your...
I think if I had to advise someone about it, maybe I wouldn't focus so much in this homogenization, because the purpose of the paper was to test the hypothesis. But instead, I would go through the different patterns for each isolated islands, because then when I saw in the other plot for the south, we have some trends. So they are becoming dissimilar, way stronger than the north. And then in the south part, there are some MPAs in the North Islands, but not in the south. So it could be that some other pressures are influencing this pattern. So when we look at the overall picture, it's kind of okay. But looking into more detail, it's not. So I think I would highlight this to take into account the specific islands, like the illegal fishing that happens there, and what are the species that they are collecting, and maybe go deep into like what are the traits that are missing, because probably we will see like a smaller size spectrum of the species there, like a lower trophic level, because they are collected. There is a fishing artisanal, but there is a fishing there. And then I think I would focus on this to help management, and then maybe not highlight that, wow, there is no homogenization. Everything is fine, because, yeah.
Good. Very pertinent.
Yeah.
Okay, we have time for one more question if there is any. Seems not to be the case. Thank you, Fernanda.
Thank you.